All right, guys, just go ahead and get started. Lewis isn't here, but he should show up eventually. All right, so the objective of today is for us to discuss key points about how to construct a manuscript, and then also we're going to discuss how to compile your contributions to the article. And I'm using the annotated bibliographies as my basis for helping you to construct your idea. So I have my own conception of what I think the directions you guys can go, but clearly you are
Professor, I think you might be having some troubles with your internet connection because, at least for me, you're frozen on the screen.
Can you guys hear me? I'm having a little problems on my main screen.
I can hear you now.
All right, I'm back. I'm gonna go ahead and project here. All right, so the very first part of this will be a breakdown of how we're going to go about in constructing the article. And so I'm going to go in and out of this presentation so I can show you live some of the things that I would like for you guys to do. For some reason, this first slide always stalls. I'm not sure why that is the case, so just bear with me here. All right, so here are the key points that we're going to talk about. We're going to discuss the development of a reference library using EndNote. So I know that some of you use Mendeley. As long as you develop your reference library so that it's compatible with EndNote, but preferably that you use EndNote, that's going to be very important as I can gather everyone's libraries of references and be able to compile the final manuscript. Another thing that we're going to talk about is developing figures, creating your own figures. So we're not going to actually show you exactly how to create a figure necessarily, but some of the key things that I want people to do as to maintain consistency. And then discuss how you obtain permission to use published figures. And then finally, we're going to discuss how we're going to actually compile your specific contributions. All right, so the very first part is developing a reference library. So I do want everyone to use a referencing software. In this case would be EndNote. Some of you do have access to this program already, and some of you, I believe you do not. And so you are welcome to come to my laboratory. I have several computers that have access to this program so that you can upload your references. And so the value of this is that it helps to preserve all of your references, and some people actually download the PDF documents into the program. I personally do not do that because I try to not keep that particular program too heavy, but for many people that's actually very valuable as far as preserving their information. Another really important thing is that the use of a referencing software allows you to very easily format your references as you put them into your Word document so that it fits the journal requirements. And so there may be some cases in which the EndNote program does not have the template that corresponds to the journal, but that's not a problem because you can Google the template that corresponds to the journal and upload it into your program. Alternatively, you can yourself create a template that corresponds to the journal if one is not located online. Another really important thing is the ability to very easily share references to another person through the use of this type of programs. And so this is very valuable because it just really helps to speed up the process when it comes time to really format the final form of the publication. All right, so I'm going to show you examples of how to use EndNote. So like I said before, I will move out of this presentation and show you live some of these things. Okay, so the very first thing is just to get you familiar with how you have the interface between, let's say, the Word document and EndNote. So let's show you as an example here. So I have my Word document open here. And so you should be able to see in the menu up here a tab that corresponds to EndNote. So here I have EndNote X7. And so what you would do is, if you don't have it open already, you would actually click on go to EndNote. And so here I went to EndNote. And so I actually have my own library that corresponds to the research that I do in my lab. And so I can show you that library. Let's see if I can open it. Let me just find it very quickly. All right, so you're gonna see basically what constitutes my library for doing papers that corresponds to my lab. And so right now I have a total of 1,709 references here. And it's actually very helpful because I can easily, you know, if I'm trying to write a really quick proposal, I can easily start compiling my proposal based on everything that I have already read. So oftentimes it's actually very helpful because as you scroll through the titles, you can recall the information that's contained within the corresponding manuscript, and that will easily help you to compile your work. All right, so I created a new EndNote file. Right now you see that it's blank so that we can be able to upload files into here. So I'm going to show you a few examples of how you can directly upload a file. You can manually type it in, and that's perfectly fine. But there are ways to just use material that's available already online. So as an example, I did a Google search here for something related to cytochrome C, and I typed in PubMed. You know, you don't actually necessarily have to type in PubMed. Often references that are available on PubMed will pop up. But I did that just for the ease of finding this information for you guys. All right, so I'm going to click on something that's available in PubMed, this manuscript here, cytochrome C functions beyond respiration. Let's pretend that we've read this article and it's an amazing article and it's definitely going to help us for our review. All right, so what we would do is we would to add this into our EndNote library. So over here, and I will say that I'm not sure how familiar you guys are with searching the literature, at least with respect to PubMed, but this overall template has changed in recent months, all right? But here you can see where it says cite, you can click on that, and then what you would do is you would click on download, and then you would click on either save or open. I would just go ahead and hit open. And what happens is that it will open it directly into your EndNote library. So my EndNote library called Cytochrome C Review. And so you see that right here. All right, so now that's already going to be saved into my library. I don't actually necessarily have to do much more than that. But I will tell you that don't trust that all the information is 100% as you want it for your final paper. So just go ahead, click on the information, and make sure that the general components are there. So here, as far as the reference type, it is a journal article, so that's immediately recognized as the bringing that file into the program. And so you want to make sure you have the general features. So here I will suggest that always be familiar with a recent publication for whatever journal you want to publish in, and so that you know what the referencing format is. But typically you want the author information, year of publication, the title, the journal information, volume, page numbers, and so forth. And so this is actually really important because it now allows you to be able to easily insert this into your document, and you know that it has the information that you want. I will say that sometimes when it inserts the reference, the existence of an alternative title can sometimes confuse the title, the recognition of the title, because something that is also very important about referencing, as you know, is that typically it is the abbreviation of the journal that is going to be go into the final document, not the whole journal name. And so the existence of the alternative journal can sometimes confuse the finding of the abbreviation, and so you can delete that from there. The EndNote program allows you to insert in abbreviations, but there's a way that you can upload a document that contains the most common abbreviations for a large set of journals. And so I'm not going to show you that here, but that's something that one can readily do. So in this case, this particular reference has the journal already in its abbreviation form. That's okay, but it's preferable to know the full journal name for your own purposes. Okay, so that's one way in which you can upload a reference from online. I'm going to show you another way. So let's say that you found a paper in a given journal publication. So for instance, here, I found this paper that's available in the ACS publications. The journal is Chemical Reviews. And so again, let's say that I read this article and I thought it was very helpful for our paper. So I want to put it into or add it into the EndNote library. So what would I do is, if you look over here to the right, and this is basically the same layout that you're going to find for the ACS journals, and you'll find something similar for publications and other publishing houses. Well, here, what I want to do is I want to export. So I'm going to click on export the RIS. And so here I have a few options. I'm going to just choose to export the citation. And so I get something that opens up just like we saw last time when we were working with an article that was available through PubMed, or at least the citation information. I'm going to just go ahead and click on OK. And so what you're going to see is that it adds it into my library, although you see only one thing that's there. But if you click on where it says all references in EndNote, if you click on there, you're going to see both of these references that I have added to the library. And again, you can click on the document that you added in, or the citation information that you added in. And so we have that information there. Okay, so we have both of these references in there. All right, so what I'm going to show you next is, if you are going to cite in your manuscript these references, let's show you how you can do that very easily. And there are a few different ways. I'm going to show you one way of doing it. So let's imagine that I want to write a profound statement, and that statement is not something that's obvious or that's trivial, that you need to have a reference to support it. So let's say that, I don't know, cytochrome C is involved in apoptosis. So that's actually a pretty general phrase. That's not something that would necessarily need a citation, but could need a citation. All right, so we write that phrase, and so what we want to do is we want to add a reference. All right, so what I would do is I make sure I put my cursor there, right after the period, or I can actually put it before the period. It depends on how the citation appears. All right, but we'll put it after the period for right now. I go into the EndNote document. I'll click on either one of these two. Okay, so I'm gonna click on the first one. And then here, what I would do is I'll click on Tools. I'll click on Cite while you write. And then I'll click on Go to Word Processor. And so when I do that, what's going to happen is that it should add, didn't quite do that just here, but let's see something here. I'm gonna try it again. It's supposed to add the citation to there. Oh, sorry. Let's redo that again. Go to Tools, go to Cite while you write, and instead of what I wrote before, not go to a word, go to Insert, select the citations. You go there, and then it will add in the citation. So I apologize for that. All right, so now what you see is that it builds up your in-text referencing, because that's what we're going to do, and that's what every publication tends to do. And it provides a number that corresponds to that reference. And so then you can go ahead and have a section already labeled as references, unless the journal asks for it to be called bibliography. Typically it's references. And so now you have that is going to build up your reference list in that order. And so here we can even add in the other reference in the same manner, and you'll add it into our manuscript. And so this is actually really helpful, because in addition to making it easy for you to cite while you're writing, okay? Because one of the worst things is to finish your manuscript or your document and then have to start adding in your references, which can be very tedious. But also this allows you to avoid duplicates, okay? And it also allows you to see what the citation is coming out to be. So in this case, I have a format that corresponds to, if you look up here in the section up here, you will see under EndNote that it has the style for Nature, all right? But we're not publishing in Nature, or at least we're not hoping to publish in Nature. We're hoping to publish in the MDPI publishing house, the journal, which is Inorganics. And so we need to make sure that we have the style that corresponds to the publication. So I'm going to go into style, and there should be a style available for the particular publication. In this case, MDPI. So I would go ahead and click on that, and it should automatically update the format so that it corresponds to the journal or to the publishing house that you're going to submit to. All right, but I will note that it is the case that sometimes journals will change their referencing format. So again, always have an example of a recent publication from the journal. So, if we're trying to publish in 2021, well, let's try to find a paper that got published in 2021 and make sure that this format that we see here matches the format from the journal. And so, and if it doesn't, well, you can do something, which is go into your EndNote, and I'm not going to go into any major detail, but you can actually edit the format of the given journal within EndNote so that it now gets updated to what is the current requirement. So up here in the tabs, you can click on Edit, and you can click on Output Styles. And in Output Styles, you can click on whatever format, so let's say MDPI. And so what's going to happen is you're going to be able to edit. So you go back into that sequence of things. You go Edit, Output Styles, and now here we're going to click on Edit MDPI. So now you have a window that opens up, and this window allows you to be able to manipulate the formatting so that it comes out to the exact format that the journal wants. I'm not going to go into how to do that because it's going to take up more time than I really want to, but if you guys want me to show you separately, I can do that. That's very easy to do. All right. So let's say that you want to publish in a journal that, for whatever reason, your particular version of EndNote does not have the template. Well, you don't really have to worry about it, because you can, as I said before, you can make your own template, although that's kind of a process. My suggestion would be if you want to do something like that, maybe take an example of a template that already exists and modify it. Another thing you can do is you can easily Google the template. So for instance, I could have Googled the template for MDPI. So let's say MDPI, EndNote template. All right, something like that. I go and search for it. All right. And make sure that
Professor, I think you're muted.
how about now?
I can hear you now.
All right, good. Thank you. Yeah, for some reason it keeps kicking me out of here. Okay. All right, so now what we're going to do is we're going to talk about developing figures here. So the really key thing about the use of figures is it adds to telling a compelling story. And so one of the things that you want to avoid is just having a manuscript, especially one that's a review article that's just very heavy on words, because you're going to lose your audience. So you want a nice balance of text and references, very key references, but also visuals that can really help to effectively say something, a key point. And so one of the things that I'm going to emphasize is maintaining a consistent style for amongst everyone. And so here I'm going to just show you how you can go about trying to maintain this consistent style, especially because we can have a sort of Frankenstein manuscript where people did whatever they wanted to, because it actually would not get published. So the quality of and the consistency of figures is actually very important. And so here I'm going to just emphasize a few key things about how we can go about maintaining this style. One of the first major ones is please pre-size your figures. All right, so I emphasize a lot of time, but I still have students that when they give me a figure, it's completely the wrong size. And so when you try to size it to the correct dimension, the visuals is awful because you lose the resolution, you can't see the text and so forth. And so I'm going to show you how you can try to avoid that problem in two different ways. But another thing I'm going to emphasize is that when you make, let's say, a plot that you want to include in this manuscript, if you want to do something like that, because remember that part of the manuscript is to provide your own analysis. So oftentimes you can take data that's available in the literature and make your own, evaluations, your correlations. And so making a plot, these are the key components that you want to make sure that you have. That you use a font that's Arial, that's common, especially in the ACS journals for the text, that the text is black, and that you're using a size as 9 point, so that it's small, but not so small that you can't see. And so as far as the plot goes, you want to make sure that the lines that you're using are black color and that the line thickness is 1 point. All right, so I'm going to show you 2 ways in which you can go about and try to preserve a consistent pre-size style, and then also to incorporate these other features that I'm mentioning to you. All right, so I'm going to go out for a moment from this presentation, and I'm actually going to use PowerPoint, because PowerPoint, believe it or not, can help you to make your figures. You can make them directly in there, and you can get some really nice resolution figures. There are some more fancy figure making applications, like for instance, Adobe Illustrator, but that one is not so user friendly. I've used it, but it depends on your abilities there. All right, so let's go ahead and open up a blank presentation. And so here we have a blank presentation. And so my suggestion then is to think about your manuscript in terms of, I would say, a traditional layout. And a traditional layout is a 1 column, so a 1 column layout, which is basically like a regular Word document. And if you notice, the MDPI Inorganics journal, well, the format is basically like that. But think of figures as being for either 1 column or for 2 columns, all right? Spread over 2 columns. And so if you're making a figure that's going to fit the size of 1 column, well, the traditional maximum width would be 3.5 inches. If you make, or 3.25 inches, correct. If you make a figure that's supposed to fit over 2 columns, then the maximum width is 7 inches. All right, and so you can use a PowerPoint document to be able to control your dimensions. And so here in PowerPoint, you can go into design and go into the slide size. So I'm going to customize mine, and I'm going to change the width so that I make something that is appropriate for a 1 column dimension. So let's make it 3.25. Okay, so I did that, and I don't really necessarily care about the height, because you can make certain things, a certain height, and then you can crop it out. But let's say that we want to make it about, maybe something that's about the same size, so a square as an example. Let's go ahead and do that. Now we have this in the dimension of a square. All right, but I will note that right now my screen is about 250 times the size that it should be. So you really want to look at this at about the correct size. And so unfortunately for me, 100% is not actually 100%, but 120 is roughly the 100% point. So you want to be able to construct your figure so you can see it, right, at the dimension that it will be in the manuscript. So that's what we're going to do here. And so I'm using my PowerPoint size regulator to be able to pre-size my figure, because I really wanted it to look exactly like it will look in the paper. All right, so that's one thing. Now, another thing is, well, let's say that we're making a plot. And so we can pre-size the plot and then ensure that that fits into our template here. So I'm going to just show you an example. And we can use Excel. And I know Excel is like, wow, why is it using such, not fancy software? But really, you don't need fancy software to make some really nice figures. The key thing is the visual that you're going to have there. So I'm just going to show you something that's a linear trend. I'm going to go ahead and plot this. And you have all taken analytical chemistry, or you will take analytical chemistry. You guys have taken lab courses, so you are probably superior than me in terms of creating figures, especially with Excel. All right, but there are a few things I want to tell you that I do not want to see, okay, so that we maintain consistency. Just pet peeves of mine, but really for consistency. Never include a chart title. Okay, your caption should be able to explain what the figure is. So we're going to go ahead and get rid of that. Never include these extra grid lines. So I'm going to go ahead and get rid of those. Okay, and another thing I'm going to do is, again, the emphasis on the size. Here, if you click on the figure, go to format, you will see that this size is randomly 3 inches height by 5 inches width. And that's definitely going to be too big, especially for, if you look at the material that we have here, we have this linear trend. It's not really that significant. This figure does not need to be this big. All right, so what we could do, we can size it. We can actually make this pretty small, but let's go ahead and make it the size of a 1-column figure. I would say that's a bit exaggerated for what is actually in this, okay? And then the height, really, we don't need it to be this big. Let's make it like 1.5, right? Something like that. That seems pretty reasonable to me. All right, so now let's apply everything else that I said to you guys as far as the coloration and the text. All right, so the text, for instance, well, let's actually add some axis titles here. Okay, so sometimes when you add the axis titles, you can see, oh, it looks a little bit squashed, and you can kind of manipulate the image so that you can do a little bit better job in terms of making it look more appropriate. All right, that seems pretty reasonable. All right, but let's do everything else that I said. So the text, right? So we want to make sure that everything is Arial, and I'm being very serious about this. Make sure everything is, okay, Arial 9, and then that the coloration is actually black, not gray. The standard is actually gray. Okay, so there we go. We do that for everything. So we go about and do Arial 9 black, okay? And so sometimes it's gonna be a little high. There we go. And then, and the more you do upfront, the better it is for you in the long term. All right, so that's one thing. All right, but that solves the font, but that does not solve the actual lines that we see here, because those lines are actually gray. So what we're going to do here, we'll click on one of the axes, and we're going to make the line so that is actually black, and that is actually 1 point. And so now we can actually see the line, and it's consistently black. Okay. All right, so that's another thing. And then the final thing is that I like to use tick marks. So I'm going to add them in. And the type of tick marks that I like to use are those that are major and that are outside, like that. And over here, like this. Okay, so that's another really important thing. And then finally, the actual plot itself, well, here are the points. I actually would like to make the points black, and then you can add a trend line. Let's, for instance, let's add this trend line, and whatever. We can just add a few more details. Doesn't really matter to me. But what I really want is that the trend line and the points, that they actually are black. The less we use color, the better. Although there are going to be points in the manuscript where the use of color is going to be important, for instance, in the crystal structures that we may want to include in your manuscript. But something like this is reasonably fine. I don't like the colors of the plot, but that's not something we're going to play around with right now. And obviously, we would title the axes, but we're not going to waste time doing that. All right, so one last thing is I do not want borders on any of the figures, so keep them like this. And then we're going to go ahead and take this figure, and we're going to put it into the PowerPoint layout. So here, I'm going to go ahead and insert it in here. And the best thing to do, unless you want to manipulate it further, is just to insert it as an actual figure and not as something coming from Excel. But you can leave it as something that's connected to Excel. It's not that big of a deal. All right, so now as you can see, this figure is already fitting the size of my pre-sized frame. And then if that's going to be my figure, obviously we're going to end up cropping this, but that's okay because we can actually crop out the other details of the height of this when we make this into an image, and you can just crop out the bottom. The key thing is that we have it in the correct size in terms of the width, and then you can play around with the height later on. That's not going to be a big deal. All right, so that helps us to control the look and as far as the use of the font and the coloration. All right, let's go back to the presentation. All right. So again, as I've emphasized a few times in emails, you can take advantage of commercial programs such as Pymol. So yesterday, Josue gave a demonstration of how to use Pymol. Also, I've given you guys access to a program called BioRender. I've actually not used myself BioRender. Students in my lab have used it. I've given you guys access to it on the condition that you use it for the purpose of this manuscript and do not share my information with other people, because I have a license that does not allow me to go beyond the purpose of a given, my own applications. But so you guys are free to use these programs. So PyMol is, freely available, BioRender through my account to make your figures. And I'm going to show you a few examples that I have in mind for you guys to do. All right, so I also get stuck on this page, so bear with me when we're getting out of this. All right, another thing is that sometimes, in addition to making your own figures, which I think is a very powerful thing, is that maybe you want to take a figure from the literature. And so that's perfectly okay. You can use published figures. What you cannot do is use it without getting permission, because everything that is published has a copyright, and so you need to get copyright permission to do so. And so don't get scared as if, oh, there's going to be such a complicated process. It actually is not. But something to keep in mind is that certain published figures, if you want to use them, they actually have a fee associated with using it in your paper or so forth. Don't worry about paying for anything. I have a budget. I can pay for certain things for this manuscript. The manuscript itself to get published, it actually has a fee associated with it. So I will deal with all of the funding issues. But just because I will deal with the funding issue, pretend that this is coming out of your pockets. And so you do not want to waste money on things that are trivial. So let's say there's a published figure that the content, the material, you can easily make your own figure in your own style. Go ahead and do that. There will be no need to get a figure that has a cost, especially if it's quite expensive. But I'm going to show you how you go about getting permission. All right, so let's exit out here for a moment. All right, so we're going to go into online. So a good example is, for instance, every journal tends to have this, but I'm looking at the journal from the European Chemical Society's publishing house. The journal is ChemMedChem. So here, what we're going to do is we're going to pretend that we're going to get permission from this journal to be able to include some of the figures. I see that there's a question. Jared, is published data like certain plots, microscope images, or anything related data need copyright permission? So the images, yes. The plots, yes. All that, yes. Now, let's say that you want to take a few data, some data from a paper, right, to make your own plot, a plot that has never been published before, because this is your own perspective of how you want to represent some information. Well, that would not need copyright information. That simply needs a citation. Like, where did you get that data from? All right, do you understand the difference there? So you do, if you in a plot, you definitely have to get the permission to be able to put that in your manuscript. Okay, so here's just an example. So here, let's say that we wanted to use some figures from this paper, although I haven't actually looked at what the figures are. What you would do is click on tools, and so other journals have their own version of this, and you're going to click on request permission. All right, so let's see. So again, tools, click on request permission. Okay, and so here it opens up to, and this is a common feature for many journals. It opens up to what is called the Copyright Clearance Center rights link. Oh, Jared, one other thing I want to mention: even if you want to publish, your own thesis, you need to get copyright permission for something that's from the literature. You just can't just copy and paste. All right, because you're actually publishing this for to get a copyright for other people to use. Where you have a little bit of liberties is like, if you're teaching a course, you can use materials that are from the literature, because we're not necessarily publishing this. You're just sharing some knowledge that was from a paper and you're conveying it to students. So that's a different story. But here, where you're trying to publish something and there's a copyright that's going to get associated, well, you need to have copyright permission from somewhere else. All right, so here, the Copyright Clearance Center. So here, there's the very first question. I would like to, what do you want? So here, there are different options. And so we're going to make a selection. So in our case, we want to use something for reuse in a journal and magazine, because we want to publish in a journal or a magazine. So what I'll do is I'll go ahead and click on that. And that opens up now a more questions that we need to answer. All right, so ready? The first one was, well, I would like to be used in a journal magazine. All right, so the requester type, well, what's the requester type? Well, basically, who are we? Well, we are a university and academic. So we go ahead and click on that. Okay, then the next thing is the reuse sponsored by or associated with a pharmaceutical or medical products company? And so the answer is no. The next thing is the format. Format is, well, the format of whatever we're going to publish. Well, in this case, we're going to publish something that's electronic, but you might want to get something that's for print and electronic just in case. There are some publications that are both. Okay, and then portion. Well, the question is, what exactly do you want to use? The full article, the text extract, figure table? In this case, we'll go for the figure table. All right. Then the next thing is, well, how many figures do we want to, of this paper, do we want to include in ours? So let's say we want to include 1. So let's go ahead and click on 1. The next thing is, will you be translating? And so no, in this case. And the next thing is circulation. Here, I always have a problem of really what to say here, because obviously we would like many people to read this, but I don't really know what is the appropriate number to put here. So I would say just be conservative, right? And just put 1 to 29, but typically there'll be a lot more people reading it. Now, the number of people citing it is not something you can easily predict. All right, so then it's to select the currency, because remember, there may be a price associated with the use of that figure. So I'm going to select the currency of the dollar. And so then what you can do is do a little quick check of the price, and it will tell you the estimate of how much it will cost. And in this case, if we want to use whatever figure in there, it actually is free, but it isn't always free. And so now you go, okay, cool, that free, that'll be fine. I'm going to get this figure, even if we don't actually end up necessarily using it. All right, so then what you do is you click on continue. And so here, we have to have an account. So what I'm going to do is I'm going to create an account that you guys can use so that we can get the permissions through that account. So I won't do this right now, because that's the next thing, is to enter the information of the account. And then what it's going to do is it's going to give you the permission. You need to download it, save that permission, and then send it to me so that I can include that when I submit the paper. I can include all of the permissions. All right, so that's basically the process that needs to be done when it comes to the use of something that has been published. All right, let's go back to the main presentation. All right, so now what we're going to do is talk about compiling the manuscript itself. Just trying to move this out the way. Okay, so very importantly for me, I'm very much a storyteller, so I want that component from you guys. And some of this can be a little bit dry, and so try to make it, if some of the things that you're reading is a little bit boring, well, try to make it, as you convey it to someone else, not boring. Okay, so that's something that's a little bit tricky to balance, but you figure out a way to capture just enough of the really important components so that this can appeal to your audience, and preferably to a broad audience. And I think that the manuscript that we're assembling could appeal to a broad audience. So as part of that, the use of words, keep the language simple, but obviously formal, and be as concise as possible. Please do not try to go for fancy words, because oftentimes you might end up just confusing what you're trying to say in the attempt to impress. So remember that there is beauty in simplicity and brevity. And think about your audience. It's an international audience. So some people may not know English that well. And for that reason, try to keep the language as simple as possible. And then another really important thing is to refer to the journal's instruction for authors. This is true for every single journal. You want to read those instructions, because they're going to tell you exactly what you need to follow. And I'm going to ask the same of you guys. Yes, I will be compiling the manuscript, and yes, I will be ultimately the one who submits this. But it's important that along the way, you guys also participate in following the instructions. And as part of this, I am going to ask you guys to compile one document with your full contribution using the journal's template. So let's exit out of here for a moment. And so what we're going to do is, excuse me, is look at the journal. So we're gonna go back to inorganics. And so if we go to the we go to the home page of inorganics. Here you're going to see on the left, and this is true of every journal, there is somewhere on the page that has the instruction for authors. Here, what you're going to see is what is this part, which is the instructions for authors. You'll click on that. And so here, then you'll get your instructions. And if you look, these are all of the instructions, okay? And so every doubt that you have, there's normally an answer here. Okay. And so here, very importantly for you guys, because remember, I want you guys to use the template from the journal. There is a template right here, which is the Microsoft Word template. Make sure that you save it, and that's what you're going to use for this journal. So let's go ahead and let's try to open this up and see what we observe. This template is actually pretty simple, but here it is, okay? And so here is the actual layout. Also, very importantly, is to also include, for those of you that have that are funding your you being a student at the whatever the RU program, what you want is the information
Profesor, se está entrecortando.
of the program, then also the grant number, okay? Because what, go ahead.
Se está you're getting caught off. Basically, I think, I don't know, for me, I can hear most of the thing you said at the beginning of discussing the article.
It's catching up now. Can you hear me?
Yes.
Okay, I'm sorry about that. I'm not sure why my laptop is All right, can you hear me? All right, can you guys hear me now?
I can hear you.
Yes.
Okay, so I'm gonna skip over the template so that you get the template is pretty much straightforward. I was just trying to emphasize a few different things that I wanted you guys to show me. Let's see.
Hello? All right, can you guys hear me?
Yes.
All righty, let's try something else here. All right, so I'm gonna use my iPad and speak through my phone. All right, so what I was just trying to tell you guys was that you can use the template in order to be able to put all the information that I need in it. So your name and any grant information that fellowship information, whether in Rice, Mark, AMP, any RU, that's really allowing you to do this type of work. That information is very valuable because it's important to acknowledge those that are contributing to your being able to engage in this, but also it also helps ensure future funding for people. All right, so that's always a good thing. All right, let's go ahead then to talk about your actual contributions. And so one of the really important things is the visual component. And so here, there are a few things that I think will be very important as far as helping to people visualize what you're saying, because I especially with this group, I know that a lot of the stuff that you were looking at was very biochemical heavy, and maybe in the terminology, especially the incorporation of how many proteins are really involved and how many molecules are involved in here, it can be a little bit overwhelming. So in that respect, you want to also make it so that it's very readable to other people. And so the use of visuals is going to be very important. So very obviously, we should include a visual that helps you capture the electron train the electron transport train, in addition to the apoptosome that actually forms as a consequence of cytochrome C interaction. So these are things that here's just an illustration of something that one could potentially make, right? But highlighting a few things that we're going to contribute to the manuscript. So just as an example, here's a representation of things that corresponds to the release of cytochrome C from the mitochondria then forming a complex with APAF1, leading to the formation of the apoptosome. To the right of that particular figure, what you see is from this particular paper are structures that have been obtained. And so they look at the formation of that actual apoptosome that look, and you can see the details that correspond to the cytochrome C and how it interacts with APOF1, and so all the components that leads to this formation. So I know that to an extent, I was speaking to Jael about helping to contribute to some of the structural details. I'm not sure if one or more of you want to contribute to the creation of some of these figures, whether it's more the cartoon illustrations that or would be something to the left, or looking at creation of figures using structures that have been uploaded into the protein database. That would be actually something that would be very helpful. So that's something that I believe would be you know, doable, and especially with the tools that we have available. So something to the left can be easily made with the use of BioRender. Something to the right can be easily made with the use of PyMol. So these are things that we have in our capability. So just for 1 second, I want to go into, let's see here. Let's see the protein database. So I don't have some of these things already open because I'm using something else. So here in the protein database, this is what we're referring to those who attended yesterday's lecture. Information is uploaded here. So here you can find a number of cytochrome C structures, for instance, and this would help be, for instance, Keisha, she's working with the substituted forms of cytochrome C with other metals. So for instance, the zinc cytochrome C structure, there are a few of them, but the zinc cytochrome C has been very helpful as far as giving information with regards to the structure and interactions of this protein. And so what you would be able to get is information that corresponds to these type of structures. So it's a little bit slow, but for instance, you get some of these structures that are available, and maybe one or more may be useful. So here's just the output of information And that's just to give you some guidance as to where you can look up some specific structures. And if you click on any one of these, they tell you what paper this information came from. Okay, and that's actually really, really valuable. And it tells you what technique was used, whether it's X-ray diffraction, whether it's some sort of microscopy or NMR, so forth. Okay, so that will be a really important component of this paper. Again, maybe one or more of you would like to contribute to the creation of figures, and again, using the tools that we have available to be able to do so. All right, so here's just my input as to from what I have gathered from you guys' annotation and how I see you guys contributing to this. So actually divided this to an extent based on the original outline that I gave you. So for instance, contributing to the evolutionary insight and the different classes of cytochrome C. Here I saw Christopher did a lot of analysis of papers that really brought insight into here. And also I spoke to Jael about some of these things that we can really be able to implement in this manuscript. So, just to give an overview, because I think providing just the general standard biochemical context of cytochrome C in terms of the different classes of cytochrome C, which classes a human one pertain to, would be very important. Give a general overview of the electron transport chain and the structures that correspond to them would be very important. But then where we can also incorporate are components that have to do with the evolution of this protein, specifically the components of it that have been conserved, whether it's the sequence components or conservation of certain types of amino acids that lead to a facilitation of the stability and also the function of this protein. And that's a very informative information that allows us to understand what is the structure of this protein that is key, the native structure that is key to optimal activity. And then if there's any possible mutants that have been identified that have perhaps greater function that maybe could be used for drug application, well, maybe we can use that information for some insight that pertains to Team 3. But, you guys are, your information will help one another in that regard. But so any information that we can get from that, we can probably apply to things that are discussed by Team 3. So that in another component of your guys' team is the role of cytochrome C in apoptosis, and there are some related functions, but specifically with regards to apoptosis. So here's where I saw Daniela, Delvin, Gladimaris, Gemily, Keisha, Glorimar, Saomi really contributing here. So I would say this is 2, and then you will see I created a 3 because I found what would be sort of an offset of some of the things I was originally focusing on. So some of the key things that I identified, and again, don't feel like you have to follow exactly how I see it. You guys are your own, artists, your own scientists, so you guys can choose how you want to implement a few things. But these are a few key points that I thought were really important. So he's just focusing on the role of the iron. Just really, what's that really fundamental function that the iron is playing, that redox capability, and then what insight is gathered from the use of non-iron substitutes. For instance, as I already mentioned, the zinc version of the protein has been used very widely to understand the structure of this protein, but also some key interactions, without necessarily facilitating the actual bioactivity. All right, so these are things that will be helpful. And so I've supplied Keisha with some additional references to help her to further flesh out this component. So another thing that I observed, and this was in things that I read for what Daniela was analyzing, discussion of reactive oxygen species generation from the mitochondria and how cytochrome C can serve as a raw scavenger and thus provide protection against oxidative stress and potentially play a role in suppressing tumor recurrence, which really applies really nicely to what will be discussed by Team 3. Generally, I saw a nice discussion of the relationship between the electron transport chain and also the mitochondrial permeability transition, that component that can lead to cytochrome C release and then therefore the onset of apoptosis, which is not necessarily the only route by which it can be triggered, but one way in which cytochrome C can be released. Another really important thing that I saw, and as discussed by Gemily and Xiaomi, was the importance of key phosphorylation in terms of controlling respiration and enabling cytoprotection. So there are some key residues on cytochrome C that the phosphorylation is actually really important, but also some key phosphorylation sites that actually inhibit the function of this protein. So that's actually another really important component that I gathered from you guys' take on the material that needs to be incorporated, in my opinion. So continuing also with the role of cytochrome C, describing factors that can suppress the bioactivity and potential utility of cytochrome C. So I saw Delvin and Glorimar really contributing here. So Delvin, I saw you had a paper about nucleotide inhibition of cytochrome C in APOF1, which is really interesting. I do want you to elaborate on what specific nucleotides are doing this, and why is this a molecular mechanism that exists, this inhibition process. And also, Glorima looking at manuscripts that focus on peroxidase and how that can potentially maybe serve as a limitation. And then this, from additional things that I've read from you guys, I saw a spin-off, which would be future uses. So Glade Maris, you focus on artificial cytochrome C. So I really want you to hone in on not necessarily some of the the methods of the formation of this. And that's to an extent important, but that's not going to be the heart of that. What I really want to see is how are these artificial C structures being applied? What are their potential uses? Another really insightful thing was cytochrome C as a diagnosis marker for certain cancers. So I saw Gemily present some information about this, but I also saw Xiaomi present some really valuable information about cytochrome C as a biomarker in the form of a danger-associated molecular pattern, so this DAMP potential property, which I think is really valuable because these could be also something that really leads in very nicely to what Team 3 will be focusing on. And then I discussed with Jael this idea of getting some structural insights of cytochrome C. Things about the charge of it, any potential glycosylation, and so forth, that can enable a better understanding of how to use it in a drug delivery system. Because understanding the biophysical properties of the protein would allow for better understanding of how you can incorporate it into a drug delivery system. All right, so these were my takes on the material that you guys provided me in your annotated bibliography. So I'm going to provide you with my assessment of your guys' assessment. And so you can take that as something from which you can build off of. And then if there are additional things that you see that I did not incorporate, feel free to include them, right? The more that we have, the better. What I don't want to see, and I emphasize this, is I don't want to see a review that's simply a regurgitation of the articles one after the other. That's laundry list writing. That's not the way to effectively write, and it's definitely not my style. I want to see you guys implementing an overview of these components that we have here and putting your own perspective on things, and then trying to create a visual that helps to illustrate these things. All right, so that was my take on it. Again, I'll provide you the presentation and also the recording, although I apologize for some of the transmission issue with respect to my laptop. Do you guys have any questions before we end?
Yes, I have a question.
Go ahead.
Should we base our future work on this outline that you just presented us?
So again, yes and no. Yes, as in like, I think that this helps to get you guys closer in terms of what you have done. And so it helps you to communicate. No, as in like, don't feel like this is, the absolute law. And if you do not follow this, you're going to get a bad grade, because there's no such thing as getting a bad grade. Okay, so don't worry about that. The thing is you're your own scientist. So if you feel like, there's something I really want to say in this, so I'm going to do it. You go ahead and do it. Okay. Again, what you guys will end up writing and what the other team will end up writing will basically be like an assembly cut. And so I'm going to be the person who will find the connective tissues, and then if there's things that I think that are missing, I can add them in and to provide that cohesion. But you guys as a team need to present something that is coherent as far as that contribution goes. Okay, so yes and no, as in like, this is not the absolute law, but this helps you to really gather your thoughts, at least the way I see it. Okay.
Thank you.
All right. Any other questions?
Sí, yo tengo una pregunta en el sentido en cuanto a nuestra referencia. ¿Es muy importante que el trabajo sea reciente, o si el artículo es bueno y pues no es muy reciente, se puede utilizar aún?
See, of course, because there are going to be some things that probably will never really be redone just because of funding purposes, or they were done so well before that that's going to be the end of it. So, and I've said this before, there are going to be some older literature that you should use in this manuscript, especially in the section that you're working on. I don't think there's, too many things that were done, let's say in 2021, but as long as there was some information that was done to give some insight about the metal, by using other metals, I think that those papers will be valuable. The goal is not to incorporate like every single paper. The goal is to incorporate key ones that help to illustrate a certain information about the metal. Okay, so again, the brevity is very important. All right, so Daniela has a question. Did you say earlier that we had to hand in a separate document for each of our contributions? No, I said one document as a team. So use a template from the MDPI, and this will be one coherent document. So there's going to be, you guys have to work together to make sure that it does appear coherent. I don't want this to be stitched together. Again, I don't want this to be like a Frankenstein thing where it's just like one paragraph and then some random other paragraph. That's not what I want. So there's going to be an element of that. It's okay if that's the rough draft, right? Because the rough draft is very soon. That's not okay for the final draft, and it's certainly not okay for the draft after the peer review that you guys are going to do with each other. Okay? All right, guys. So if you do have any additional questions, just feel free to email me. I will provide you with all this information. And so have a good day and take care. Enjoy your weekend.
Thank you very much. Have a nice weekend.
Bye-bye.
Gracias.
Professor, I wanted to talk to you very briefly about something about the structural insight of cytochrome C, the drug delivery system. So I wanted to specify, what did you have in mind in terms of what type of drug is being delivered? Are you trying to, because there are different, different
the drug
approaches.
The drug being cytochrome C. So
okay, yeah, okay.
Right. So where I find that a missing element from everything that I've read, right, is really just some of the fine details of the protein itself, which I think that that you can help with, like, so we have a general idea of the size, but not so much about some of just the general components. So Christopher did have things about conserved sequences, whether it's the actual conservation of the amino acids or just the components of it, but just some additional features that would probably be helpful for us to be able to give insight to the other team. So that's what I mean. And also, if you're interested in contributing to making the figures, especially some of those bigger structures, you're more used to seeing like these huge supermolecular structures. So maybe that's something that you can help with. Josue can also help you to construct some of these things. Okay.
Okay. Perfect. Thank you.
All right. You're welcome.